In the past few weeks, we've talked about the various causes of dyspepsia, and we've talked about how normally the stomach is protected um, from the corrosive effects of the gastric acid by the lining, the mucus lining that is here depicted in blue. Now, the mucus secretions in the esophagus are much less extensive and have much less extensive capacity to buffer the effects of the stomach acid, which is why reflux can cause problems. And we've also discussed why ulceration can take place when the protective lining in the stomach is damaged, which can occur either by Helicobacter pylori infections or by the use of NSAIDs. So in this lecture, I'm going to talk about the various therapeutics and their interaction with biological targets. So here are the various therapeutics that are used to treat dyspepsia. And for completion, I included here antacids and alginates, although I will not talk about them in much detail. And the reason for that is that their therapeutic effect is really mediated by physicochemical mechanisms. So their therapeutic effect is not mediated via interactions with biological targets, and for that reason I've just included them for completion. Now in the first part of this podcast I will talk about H2 receptor antagonists, which is an example of a therapeutic targeting a G protein coupled receptor. In part 2 I'll talk about proton pump inhibitors, which target a protein involved in transmembrane transport, which we've discussed extensively in lecture 2. And finally, part 3, I will talk about NSAIDs, which can of course cause gastric ulceration and their interactions with their biological targets. So first we will discuss the effects of H2 receptor antagonists. So here is a schematic diagram of the H2 receptor, which is a G protein coupled receptor, so it's characterized by these 7 transmembrane helices. And the normal ligand that activates this receptor is histamine, and this is an example of an H2 receptor antagonist, ranitidine in this case. Now there's not much or there's no structural information about how these molecules actually bind to this receptor, but based on the general examples that we discussed in lecture 2, we can make a model and we can imagine how these molecules bind to the H2 receptor. And so we've seen in lecture 2 that the 7 transmembrane helices can form a sort of barrel-like shape, and on the inside of the barrel the ligand will bind, whether it's an agonist, an activating molecule, or an antagonist. Next slide shows an example. So here is a model of the H2 receptor, or at least part of the H2 receptor, which is made based on its similarity. And in green, highlighted here, and here's an enlargement, are 3 amino acids that people have found to be important for the binding of histamine. And as you can see, they are localized here in this sort of in these transmembrane helices, which is in agreement with the examples, the other examples that we've seen in lecture 2. Now, if we look at these amino acids here in green in more detail, we find that there are 2 aspartates with a carboxylic group and a threonine side chain involved in or proposed to be involved in the binding of histamine. So here's an ionic interaction, and there's 2 hydrogen bonds. And so this is a model for how histamine normally binds to the H2 receptor, and this would lead to an active receptor and active signaling. So presumably ranitidine or other H2 receptor antagonists would bind in a similar region. And the question is how binding of an agonist or antagonist in the same region can result in the one case in an active receptor that can interact with the G proteins, ultimately resulting in the production of cyclic AMP, whereas in the case of an antagonist binding in the same region, there is no binding to G proteins and the production of cyclic AMP. So this can be understood by looking at another receptor, which is next. So in this slide, you see two models of a G protein coupled receptor. It's not the H2 receptor, but a very related receptor that helps us to understand how an agonist can result, binding of an agonist can result in binding of a G alpha protein, which ultimately results in a production of cyclic AMP, whereas binding of an antagonist here on the right does not lead to production of cyclic AMP because this receptor cannot bind to the G alpha protein. So how is that? How does that work? So overall, you can see that. So this is the active receptor bound to an agonist, and you can see that overall, if you compare this structure with the one here on the right, which is inactive, that overall the structure is really very similar. However, if you look closely, there are many differences which are caused by the different binding of the antagonist versus the agonist. And the most pronounced difference is here on the cytoplasmic side of the receptor, and it's circled here in red. So the different binding mode of the agonist versus the antagonist results in the case of an agonist in this alpha helix, which is a binding platform for the G alpha protein, which is now activated, and this ultimately leads to the production of cyclic AMP. And you can see that in the presence of an antagonist, the many different subtle changes in the structure lead to the absence of this alpha helix on the cytoplasmic side of the receptor, and hence the inactive receptor cannot bind to the G alpha protein and does not result in the formation of cyclic AMP and active signaling by the H2 receptor antagonist. So this provides a way that helps you to understand how binding of an agonist versus antagonist results in different conformations of the protein that are either active or inactive.